about some research using stem cells that we've generated over the last year and a half. But before I do that, I want to do a primer on how you move when you want to. So if I want to move my right toe, what we know is that an impulse starts in the left side of the brain, in a nerve, also called a neuron. And that impulse carries down through the brain stem, crosses over in the spinal cord, and goes down to another neuron called a motor neuron, a ventral motor neuron. So an impulse is started in one side of the brain, in a neuron, travels down that neuron towards the other side of the spinal cord, interacts with a second neuron called a ventral motor neuron, which then leaves the spinal cord and goes out towards muscle. That electrical impulse is carried out in that ventral motor neuron, where it then gets to the muscle, and the muscle contracts and the toe moves. Okay? So that's how we generate volitional, voluntary movement. Now, some of you don't have that so well. And so there are some mechanisms whereby that won't occur. And we've heard some of them yesterday. For example, demyelination. So myelin is the insulation that wraps itself around the neuron. The axon is another term that we've used a lot. So the neuron is a small little cell with incredibly long axon or process. And that needs to be wrapped with myelin in order to conduct that impulse that I was talking about well. So in demyelination, the impulse sometimes gets short-circuited. So it doesn't arrive. So I say, I want to move my right toe, but it doesn't get there. Somewhere along the way, that impulse gets lost. And in many cases in transverse myelitis, it gets down to the level of the injury, the focal inflammatory process in the spinal cord, but doesn't get any further. And in some cases, that's because the myelin is gone. And so the impulse short circuits. It's just like an electrical wire in which you need conduction around the outside. And if you lose that, the electrical impulse short circuits. Now there's a second mechanism for failure of movement when you want to. And that's neuronal loss. So that means we're not talking myelin. That means that the neuron itself is gone. And that's what I want to get at a little bit today, because we know that that occurs. In fact, we think in most people with transverse myelitis, both mechanisms occur. And you heard from Jeff Kosis and John McDonald about some very exciting things with remyelination. It's probably easier to envision remyelinating than it is to restore neurons. And so it makes sense that that will be the first to be attempted. Motor neuron regeneration is going to be more difficult, and it's going to take more time. So, I will, if I start saying jargon and things that people don't understand, just raise your hand and I'll back up a second, okay? Now, some of these I will skip over, or I'm just going to gloss over because I want the scientists to see them, but I'm not going to dwell on them too much. Essentially, what we have done is we create paralysis in a rat by giving it a virus. And the virus goes to the spinal cord and paralyzes the animal. So we're very interested in that process. But we're also interested now in restoring function to that rat. So both arms are interesting to us. But what I want to tell you about now is once the rat is paralyzed, once it's cleared the virus, now what? Can we restore function to that animal? It's called Synbis virus. You probably have never heard of it. You've heard of its cousins, though. Its cousins are West Nile virus and Eastern equine encephalitis. I bet you're probably more likely to have heard West Nile virus. It is a virus that infects neurons. And so when we give it into this animal, it infects neurons in the spinal cord and kills them. So it is the ventral motor neuron up and down the spinal cord that die as a result of this viral infection. And this is what the little guys look like. So if I have a, do we have a red? So this guy's not normal because he's got this abnormal spinal curvature. It's called a kyphoscoliosis. And that's reflective of weakness of the muscles in his back. And so they pull the back in different directions and he gets this abnormal curve. And this one is a later example following infection with this virus. And this animal is paralyzed. It's got a lot of atrophy in which the muscles just kind of disappear and the hind limbs are splayed out behind it and it can't walk with those hind limbs. There is relative preservation of forelimb strength in these animals. So they can crawl around, and you'll see videos of them. They can crawl around on their forelimbs. It's their hind limbs that aren't doing so well. This is just a trick that we can put a green fluorescent protein, a jellyfish protein, onto the virus and say, now we want to know where the virus goes. And we don't have to do very sophisticated things. We just look in the spinal cord what cells become green. And so this is clearly a green one. And what cell is this? Well, we know this is a ventral motor neuron. So this is the neuron that's at multiple levels of the spinal cord, sends an axon out to the muscle, allowing you to move. These are more ventral motor neurons. You've seen pictures of these throughout the weekend. They're basically the biggest cells in the central nervous system. And in a rat that's had this paralysis, there's many, many fewer of them. This guy is normal. Well, he was before we killed him. And this guy has got many, many fewer motor neurons. This is a silver stain looking at axons. Remember, axons are the long processes that come out, and we can actually look at those axons, and we can say, okay, are they there or are they dying? And in this case, what you're looking at here, DR stands for dorsal root. It means sensory nerves. We're not really talking much about sensory nerves, but sensory nerves start in the skin or muscle, and they relay impulses from the skin or muscle to the spinal cord and up to the brain. Okay? So it's a different system that runs in parallel, but actually a different direction from the motor system, which starts in the brain, goes down to muscle. And in this particular system, the dorsal roots, the sensory nerves, are really, really spared. So anytime you see a little dot here, that means an axon coming right out at you, out of the screen, and there's no damage here. This is all normal. Whereas you see bad stuff happening here. And this is the ventral root, and these are the motor axons. In other words, the motor nerves send their axons out, and they're not doing so well. In fact, about 92% of them are dead. And many of you may have gotten EMGs, electromyograms. And it's a needle is stuck into the muscle. And what we do is we can look at the muscle and see if it's lost the neuron that should be there, that should be coming towards it. And if that neuron is gone, it has a characteristic abnormality, and the nerve conduction studies are abnormal. These are electrical tests. And essentially, I just want you to look at the bottom one if you can see it, although it's pretty low. It's a flat line. And we're trying to induce a motor action potential in this rat. It's paralyzed, and we can't get one, so the axons are gone. And then the muscle itself is very characteristic of muscle which has lost a motor axon to it. Even the muscle itself, when we take a biopsy of the muscle, shows characteristic features for the clinicians of neurogenic atrophy with angulated fibers, and you see the muscle fibers themselves here viewed in cross-section. We've just kind of taken a little chunk of muscle, and the muscle fibers atrophy. And you know that occurs because in people who have transverse myelitis and motor neuron disease, there is a lot of atrophy, and that's why the muscles start to just shrink, and that's what atrophy means. So this is really a pure lower motor neuron paralysis, and we have looked at stem cells as a potential therapeutic strategy to restore function to these animals. This is what we do. You're looking at a schematic of an animal from the side. This is the animal's brain. That part right there, does anybody know what part of the brain that is? cerebellum, and then this is the spinal cord. And so essentially what we've done is we do a spinal tap, but instead of taking fluid from the rat, we administer cells. And I'm going to tell you what cells we used. And we've done a lot of work with this, and as John and Jeff showed you yesterday, it's very critical that we very clearly define the cells with multiple labeling techniques. In other words, we've got to be able to tell which are our cells that we've implanted and which ones are there from the beginning. And I'll show you some pictures. Now, where do we get these cells? These are from human fetuses. Now, I'll tell you that the cells that we used were derived from a human elective abortion in the mid-1980s, one, and the cells have been grown and expanded ever since without any need to go back to another source of fetal tissue. And these cells, through all of that, retain their ability to be stem cells. And so they have been expanded in numbers to the point where we could easily conceive of, from that one abortion, 500,000 therapeutic interventions for patients. So it would be more complex, to me anyway, but this is just my view, if we had to have a recurrent source of fetal tissue, and I think that that may not be the case. We all want to explore even non-fetal sources. Right now we're not quite there. So these come from the genital ridge. These are cells which in that fetus were destined to become gonads, oocytes and sperm. And what we do is we take them and we've grown them in culture through a whole series of things. We add some growth factors and we remove some growth factors and we do a whole bunch of things, ending up with cells in a dish that look pretty bland. They don't look like anything in particular, but they have the potential to differentiate down towards multiple lineages. So you can shunt these cells towards becoming cartilage, or towards becoming bone, or towards becoming red blood cells, white blood cells, things like that. And of course what we're interested in is, can we shunt them down towards becoming neurons, oligodendrocytes, and astrocytes? Those are the three biggies in the central nervous system. And so some of these will come out, some of these won't. What you're looking at here is a cross-section of the spinal cord. And in this particular thing, anytime you see a green cell, that is a stem cell, which we had labeled prior to implantation. Because remember, we've got to define which ones are the stem cells and which ones are the host cells that were there. So anytime something is green here, this is BRDU, it means a stem cell. And you can't see it too well, but this is the outside of the spinal cord, and this is spinal fluid. Okay, so we actually did a lumbar puncture. We injected the cells. The first question is, do they migrate into the spinal cord? Because they're not going to do us much good if they stay out here in the spinal fluid. And so what we can see here, and it was a dramatic finding for us, is that many of the cells do migrate into the spinal cord and actually go to the ventral horn. This is the area where the motor neurons are, or were in this case. So in our animal model, the motor neurons are dead. And so there's something about the death in the ventral horn of these animals that attracts the stem cells to that particular region, which is very exciting to us. And many of the other researchers in stem cells have found this, that the death, the injury, is an attractant for the stem cells. And the reason it's so exciting is because we may not even need to be that smart, because they'll go anyway. Now, we could probably improve the efficiency of that, but there's something almost magical about this that attracts stem cells to that site. Other labeling, such as Herx as a nuclear dye, which just convinced us that this is real and that these cells get into the spinal cord. More of these. I won't belabor this. And so the next question is, okay, they get in, fine, but do they do anything? Do they turn into neurons, for example? Because we know that we need to have neurons. And what you look at here is, when in this particular slide, any blue cell is a stem cell. So we had given it this blue dye prior to implantation. Any green cell is a neuron, or is a cell that is expressing something called Neuron, which defines it as a mature neuron. And can you see this cell down here? It's green, but it's not blue. So it is host. It is just a neuron that survived and is in that spinal cord, and we've got it in this section. You see this cell here? It's blue, so it's a stem cell, but it didn't differentiate into a neuron. It's just sitting there hanging out. This one here is both. It's blue and green. Here's another one, blue and green. So that what we've shown is that these cells tend to differentiate into neurons in this system. Not all of them. In fact, most of them don't. We only get 6 to 8% of the cells differentiating into neurons. And 6 to 8. Now, we'd like to get that better, and we probably will be able to, but right now we're not smart enough. Here's what it looks like, just for those of you who are inclined to tables. 64% of them actually remain around the outside of the spinal cord. 36% of them get into the spinal cord. Now, of those that are just outside and didn't quite do the thing that they should have done, none of them turn into neurons. Of those that get in, depends on the area, 18% of those cells are going to the ventral gray matter, which is where the motor neurons are dying. Much fewer of them go to other regions, so they really are kind of drawn towards the area where the cells are dying. And another interesting point is that differentiation into neurons really only occurs if they migrate to the right area. So there's so neurofilament positivity in Neuron, which is a marker of mature neurons, 6% of the cells that we looked at and 8% of the cells in the ventral gray matter. But none of them in other areas in the spinal cord. So this death of cells is not only an attractant for them to come to this area, but then once they get there, they're more likely to turn into neurons. Okay, so what happens to the animals? I've shown you that they get into the spinal cord, and I've shown you that some of them, though not most of them, turn into cells that look like to us neurons. And here are bar graphs, and if you don't give them cells, or you give them stem cells that you've killed prior to implantation, or if you give them non-stem cells, or if you just surgically treat them but don't give any cells at all, nobody gets any better. All the animals remain completely paralyzed. And this was the exciting part for us, is that when we gave them these human fetal cells, at 4 weeks we saw nothing. At 8 weeks we essentially saw nothing. But at 12 weeks, they started moving. Now, we didn't know which was which. We were all completely blinded to this because we don't trust ourselves. And so it's all coded. And so, but at 12 weeks, we start to see some animals moving. We don't know if it's the control or the experimental group. And so when we then kind of broke the code later, we were quite excited to see that all of the animals that recovered had gotten the stem cells. So let's do some video here. Okay. Just like John's mice, they just weren't motivated, I think. All right, so he's doing okay, except he's got no movement of his hind limbs. His forelimbs are okay. He can move around. We put a little Mary Jane wrapper there to see if he'd become interested in that. Okay, and here is an animal that has gotten stem cells and is doing better. So he's now got his hind limbs under him a little bit, as opposed to being splayed out behind him, and will now take a few steps once we can get him to, okay? So a few steps there. So doing pretty well. He actually cannot bear weight very effectively. And when he does, you can kind of see that he collapses. So the hind limb kind of falls out behind him. So he brought it under him and tried to push off, but then as he tried to push off bearing weight on that, he would collapse. And this is the average score that we got in all of the animals, 21 animals in each group. There was one animal that did much better than that. And I tell you, it's only one animal, so 1 out of 21. None of the others look this good. But he looks pretty good. He's still got this curvature of the spine. So, why did they get better? So, there are really two possible scenarios, at least two, that we can imagine. One is that the picture that I've been trying to paint is that they've turned into neurons, and where the motor neurons are dead in the animal because of the virus, we now give them cells that turn into neurons and send their processes out to the muscle, and all is well. The other possibility is that these cells get into the spinal cord and serve to enhance the function of those remaining neurons that were there to begin with. So there are 8% of motor neurons in that animal are there to begin with. Now, we've heard from other speakers, and what we know from humans and animal studies is that you don't need a whole lot of neurons to move a limb. In fact, some people say even 10 or 20% of the normal number of motor neurons is required to move the limb. And yet our animals could not move at all. And so you can imagine that maybe a simpler task would be to enhance the function of those remaining 8% of neurons that are there could result in that. And the answer is that both of those mechanisms occur. I'll just show you that we want the BBB score is the one that John used and the one that we've used, but we've also used some independent measures to show that those animals get better. I think you can see that they get better, but the other thing that we do is we put them on their back and we ask them to roll over, and we time them, how long it takes for them to roll over. And we think that if they roll over more quickly, they're probably stronger. And in the stem cell treated animals, after 12 weeks, it takes them a lot less time to do that than it did at the beginning. Whereas if we do all of the control experiments, there's no difference. Whatever time it took that animal to turn over at time 0 is the same time that it took for that animal to turn over at time 12 weeks. Okay, so in this particular photomicrograph, blue cell defines a stem cell. So what we did is we took these animals that had recovered at 12 weeks, and we put a chemical dye, a fluorescently conjugated dextran, into the sciatic nerve. And we asked that fluorescent chemical to be transported back to the spinal cord in that animal. And we asked the question, are any of those cells that had their processes out in the sciatic nerve stem cell in origin, or are they all host? And so, because that's the ultimate here, is if a motor neuron will send a process, if a stem cell will not only go to the right place, but also differentiate into a neuron and send a process out to the muscle, boy, that's the holy grail. And so what you've got here is anything that's green is a cell which had its process out in the periphery as defined by this fluorescent chemical. And so you see 3, although it's not showing up all that well here. I wonder if we can shut the lights down a little bit further. So there are 4 here that had their processes out in the periphery. One of them is a stem cell, and the others actually are host. By other factors, we can say, well, this is a neuron. It looks like a neuron. It's huge like a neuron. Remember, these are much bigger than really any other neuron in the brain or spinal cord. And so it's about 40 micrometers, which is characteristic of a motor neuron. And this is the overlay of those. So, rarely, but it does occur, which was really a stunning thing for us, these cells send their processes out into the periphery as defined by a retrograde labeling. Now, when we first showed this to people and submitted this, they said, no. What this is this is a stem cell sitting on top of a motor neuron, and you're being fooled. So we did confocal microscopy. Basically, it just means we're taking a very, very thin section, eliminating the chance of anything sitting on top of. And again, green means that it had its axons out in the periphery. Really, 3 cells here had their axons out in the periphery. The red defines neurons. So there are 4 neurons here. This one actually did not send its process out in the periphery, or at least we couldn't detect that. Blue defines stem cell that we had labeled. So this one here is a motor neuron that had sent its process out into the periphery and is stem cell in origin, whereas the others are host. This one is a stem cell but didn't send its process out into the periphery, and you can also see some stem cells that are just hanging out and didn't do anything. Now, the other mechanism also occurs, and in fact, that which I just showed you here is rare. We don't have that working very well. On the order of 1% of the retrograde labeled cells are stem cell in origin. So I truthfully am excited about that, but I don't think that's why the animals are walking. But what we know is we can take spinal cord sections. So, and this is a spinal cord section in a dish where all the cells are alive and we're giving them growth factors and nutrients, and we can put the stem cells outside of that, called spinal cord organotypic, and we can say, if we put these stem cells outside of this spinal cord culture, what happens? And what you see is that the axons from the motor neurons, these are motor neurons here, are very, very happy to grow out to and invade within, here at higher power, this cluster of stem cells. So there's something that the stem cells are secreting which is very, very attractive towards motor neurons. And we know that they're secreting some things like BDNF, and there are probably some others, although we haven't really defined that as well. I think that's it. So human fetal stem cells distribute widely when given into the spinal fluid. They migrate from the spinal fluid into the spinal cord. Some of them, although a minority, differentiate into cells that express mature neuronal markers. They occasionally send out processes to the periphery in the sciatic nerve. They also provide tropic and trophic support to remaining host neurons so as to enhance their function, and the animals get better. I won't do this yet. David Arani is here. Lots of people have helped with this. And you've seen this before, but I've got to show it again. Thank you. That's it.